Hello, everyone, wherever you are. Welcome to this webinar. I'm Kordo Saeed. I'm a consultant clinical microbiologist in University Hospital Southampton, an honorary associate professor of microbiology at Southampton University in the UK. It's my delight to introduce this webinar on behalf of the Rapid Diagnostics and Biomarker Working Group of the International Society of Antimicrobial Chemotherapy. ISAC was founded in 1961 and is a federation of affiliated member societies. The aim of ISAC mainly is to increase knowledge of antimicrobial chemotherapy and combat antimicrobial resistance globally. ISAC has a global membership of around 60,000 individual members belonging to over 100 national and regional societies. And it hosts International Congress of Antimicrobial Chemotherapy meeting every second year, so the ICC, and this year would be held in Istanbul in November 2024. If you have the chance, it would be a great opportunity for learning, sharing knowledge, as well as networking. Today, our webinar is from the Rapid Diagnostics and the Biomarker Working Group, and I'm delighted we have three excellent speakers with three very exciting talks related to the subject of diagnostics in infectious diseases and microbiology. With that, I am delighted to introduce the first speaker soon. But before that, please, if you have any questions, please put it in the chat as a Q&A, and we try to address the questions after each talks. Also, this presentation would be recorded, so it will be available to you in due course by ISAC, subject to agreement from all the speakers. So I'm delighted now to introduce Dr. Lucy Bock, who is has got a PhD in molecular medicine from the European Institute of Oncology in Milan, Italy. Lucy currently is a senior project team leader in the antimicrobial discovery, development and diagnostic group at the UK Health Security Agency in Porton Down. Her interest lies in converting scientific knowledge about antimicrobial resistance into real-life solutions. And for the last 3 years, she's been working on developing a rapid diagnostics of antimicrobial sensitivity from UTI on IFAS. So I would let Lucy to start her presentation, and then looking forward to it.
So my disclaimers. Why do we actually need a rapid urinary tract infection antimicrobial susceptibility test? Well, first of all, because of the number of antibiotics that are being prescribed. So here's just an overview, for example, in Switzerland, but it's the same in most countries worldwide, or at least developed countries, that for urinary tract infections is the number of antibiotics that is most prescribed. Then also the number of samples that are processed. So, for example, we've been working at the University Hospital Southampton, where they sample about 300 urine samples a day. So obviously, if that could be streamlined, speeded up, and made easier in general, that would save a lot of time, money, and also then obviously have patient benefits. And also overall, actually, urinary tract infection can lead to mortality as well as morbidity. So it can lead to bacteremia, and here's a recent publication showing that the mortality is actually being increased. So what we've been using technology is bacterial impedance cytometry to speed up UTI ASTs. So the basis of it all was that currently to check for antimicrobial susceptibility of isolates causing UTIs takes quite a while. So we have, hope you can see my pointer. The urine culture is plated, and we need an overnight incubation. We then dilute it to a certain concentration, so if we can, the bacteria, so we can expose to a fixed concentration of antibiotics. That again has to be incubated overnight, so that the following day we can then see which bacterial strains are susceptible or resistant to the antibiotics that we want to treat with. So overall, we're talking 2 days. That's of course rather long. So the aim of the project that we were working on was to develop a phenotypic UTI AST. So we want to stay with the phenotypic, but reduce this time from 48 hours to ultimately less than a shift, so less than 8 hours. We were aiming to develop this at the same cost and accuracy as the current gold standard. And to do this, we involved all stakeholders. So microbiologists, which was us at UKHSA, the engineers at the University of Southampton, the biomedical scientists who would actually be using this, so at the hospital. Then we had some involvement from public patient involvement, so to give some feedback on how this could actually affect patients and their carers. The actual clinicians like Kordo, so how they would actually act on these results, what would change, what would be important to look at. We also had some healthcare economists involved to check what economic benefits there would be of introducing this fast phenotypic test. And part of the grant was also to found a spin-out company, which we did, called iFast. As I said, we're now not linked to that directly now. So the aim was to go from this 48-hour process to less than 8 hours by using culture plate. We again plate it onto a purity plate, but only for 2 hours. This is mainly so that we have logarithmically growing cells, and also to remove any kind of inhibitors that might have been in the patient. We dilute again to the same concentration so that we can use the current UCAS breakpoints. We expose to antibiotics and also have 2 controls that are not exposed to any antibiotics so we can compare the changes. We expose only for 2 hours, and then we read it on this iFast machine, which is also very quick. So what is this iFast machine? Well, it's based on the technology called bacterial impedance cytometry. The idea behind it is that we have a bacterial cell, and this bacterial cell will impede a current that flows through it. Because of its size or its membrane makeup, that impedance will change. So basically the current can no longer flow properly if we have a bacterial cell that interrupts this current. So we look at 2 different frequencies, 5 and 40 megahertz, and then we can see for each individual cell how this current has been impeded. And using some clever calculations, we can from that work out the electrical diameter of each cell and the electrical opacity and phase of each cell. So that means that if we have, for example, here, a Klebsiella pneumoniae population that has not been exposed to an antibiotic, it would have an electrical diameter of about 2 and an electrical opacity of about 0.75. We always include some polystyrene beads so we can do some normalization. If this population has now been exposed to, for example, a beta-lactam, which of course affects the membrane, that means that the impedance will change because the membrane will have been affected and the cells can swell. Therefore, the readout of these cells in this population will have shifted. So it has a higher electrical diameter and a lower opacity. So that is one readout, comparing the change in diameter and opacity from those that haven't been exposed, so the control, to those that have been exposed. Alternatively, here again, we have no exposure, and then we have this particular strain exposed to trimethoprim, so it's resistant to trimethoprim, so you can see there's no change in the population comparing the control to the trimethoprim exposed. However, it is susceptible to ciprofloxacin at this particular concentration, so you can see that the population has disappeared. So there are various different metrics we can look at. We can look at just a cell count, whether it has changed or whether it has decreased, but also we can look at the shifts. So the first thing we did was to have a look at a proof of principle, basically in the laboratory under controlled conditions. So again, the protocol was to plate, dilute to a particular concentration, expose to breakpoint concentrations of antibiotics that are used currently at Southampton for treatment of UTIs, incubate for only 2 hours, and then read it on the iFast. So looking at basically the impedance changes. Looked at 57 strains. We concentrated on E. coli and Klebsiella as they're the main causative organisms for UTIs. The strains we chose had varying MICs and resistance mechanisms to these antibiotics, which are the ones we tested, so different classes of antibiotics. And we used the UCAS breakdown for Antibacteriales from 2022, which is when we started. And we chose to look at the susceptibility breakpoint so that we would have a rule-in test. We can treat with this. The strains were a mix of reference, clinical, and laboratory strains. And we always compared them in parallel to the gold standard, so the microbroth dilution MIC. So as you know, the microbroth dilution MIC has an area of technical uncertainty of 1 doubling dilution. So if we have a non-concordant result, we don't know whether it's due to the IFAST, where we're only reading breakpoint concentrations, or the microbroth dilution, where we're reading MIC concentrations. I have removed those strains that have an MIC that is 1 doubling on the breakpoint or 1 doubling dilution way. So if I do that, we wanted to know which metric would be the best one to look at the concordance and between the gold standard and our new method. So we looked at, as I showed earlier, the cell count, but also the coefficient of variance of diameter, opacity, and phase, and also of the median diameter, opacity, and phase for each antibiotic. And here we have the results in ROC curves. So basically, this dotted line means that you might as well throw a coin to see whether they would match. If they go all the way to the top here, that means they have 100% concordance. So you can see here for these 8 antibiotics that using the cell count metric, and there are some others underneath it as well, for some of them, we reach 100% concordance between the IFAST method, which is measuring just breakpoint exposure, and the MICs that are currently used in the microbroth dilution. This is when the area of technicity has been removed. The only exception is nitrofurantoin, where actually the median phase is the better readout, and we don't quite reach 100%, but we're still very high concordance. So then, actually, while this was already going on, a PhD student, Bethany Martin, she went into the hospital to have a look whether this would potentially work on clinical isolates. So she followed the same protocol, but it was still a manual system, so not this nice automated box. It was still very manual. She had a look at 10 samples against the 8 antibiotics, and she only used samples that were confirmed to be E. coli. And in the results, she actually showed that 79 out of 80 matched the current diagnostic laboratory AST results. So we were very happy with that. So we then proceeded to set up a much bigger concordance study, which only ended last week at the end of March. So these results are rather preliminary. So the setup was basically the urine sample was confirmed to be an infection using a microscopy. The purity plate from that, it was confirmed to be E. coli or Klebsiella. That's what we concentrated on initially to match it with the proof of principle data from the laboratory. We then plated on 2 hours on another plate, and then basically followed the same procedure. Dilute into saline. Using the IFAST, we could do a very quick cell count rather than having to rely on McFarland standards. We then added it to the media, added it to freeze-dried antibiotics at concentrations that was a breakpoint concentration of the same antibiotics, and half breakpoint concentrations, incubated for only 2 hours at 37 degrees, diluted it into saline, and then read it on the IFAST. For each sample, we only take 30 seconds to read on the IFAST. So this trial was actually conducted by medical scientists, so not by us who have been messing around with the technology and know all its quirks. So we wanted to know, how accessible it was for somebody who had to process samples day in, day out. We used, as I say, 2 antibiotic concentrations based on the fact that we were comparing breakpoint concentrations to MICs. So we wanted an additional concentration. We only looked at E. coli and Klebsiella initially, but I haven't put the data here because we're still working through it, but we did also then actually go blind from just urine, so not from purity plates. We literally just confirmed using microscopy that there was an infection, and then took the urine and incubated for 2 hours, and then on a plate, then transferred to the antibiotic exposure, and then read on the IFAST, which also looks promising. I'm happy to answer questions about that, but I don't have the data here yet because it's preliminary. So the preliminary results are that we have a 94% agreement across the 107 isolates that we looked at with the 8 antibiotics. That breaks down variously depending on the antibiotic. That is because of the distribution of susceptibility resistance we have to various antibiotics, and also a few others. So some of these errors, 76% of them were major errors, 24 were minor errors. And some of these errors are caused by differences in concentration of antibiotics that we were testing. So, for example, we were testing amoxicillin, whereas the system in the hospital was ampicillin. We had different breakpoints for co-amoxiclav, and we tested the susceptibility breakpoint, whereas the laboratory was testing resistance breakpoint. So we did expect some discordances, and we are going to investigate those further just to confirm them. Also, of course, the expert rules were already applied to the lab results. So, for example, that if it's resistant to amoxicillin, it has to be resistant to co-amoxiclav. So if there was discordance, we can't really check that. And the other thing is, like I said, we are looking at all these different metrics. And for example, nitrofurantoin wasn't even in the preliminary, was not perfectly matching. So we're still looking at using now these actual results. We're having a look at whether other metrics would give us better concordance. So just to say that we have also used this for other applications. So for example, Bethany also had a look at MRSA determination, so ever so slightly modified protocol. This is, for example, what a Staph aureus looks like. You can see it has a very different signature, so different organisms look different. And this is what a susceptible one looks like, and a resistant one. It has this stretch. And she looked at 60 clinical isolates, and she had 100% concordance, but of course had it 24 hours earlier than the current testing. So that could have a high impact also. We also had a brilliant student, Caitlin Daniels, who was looking at Neisseria gonorrhea AST, which is also a pain to grow in the laboratory. So ever so slightly changed again, just growing it up for 30 minutes from a plate, and then again exposing for 4 hours. She looked at 6 reference strains and tested against 3 antibiotics, and she got AST results in less than 8 hours for a purity plate. Currently, that can sometimes take days. Again, you can see that Neisseria looks different again compared to other organisms. These are susceptibility ones, and for those that she tested, she got 100% concordance, looking at the decrease in cell count. One thing we're also looking at currently is using it for phage. So here's some data showing that, for example, this strain of Acinetobacter baumannii was susceptible to the low-key phage, and you can see this is untreated, not treated with phage, and this is after being treated for 2 hours to increasing concentration of phage, still susceptible, whereas this is actually a resistant strain, and you can see that at the lower concentrations of phage, it is indeed resistant after 2 hours. So that's a much faster way of doing AST than currently it takes, well, definitely overnight. And this also, we're rather pleased of. Here we have a controlled strain that's not being exposed to a phage. This one has been an E. coli strain exposed to E. coli T7 phage, of which we know that it has a 17-minute cycle time. So looking every 5 minutes after exposure, you can see for the control that there is no change. However, for the one that is exposed to phage, you can see nothing for 10 minutes. After 50 minutes, there's an indication here there's some dead cells. In fact, after 20 minutes, the cells have basically all died, implying that within 20 minutes, we can very clearly follow this infection process. So that gives us a greater understanding of the phage infection process. It's also much faster, like I say, and may also help us to test for resistance development. Another thing we have noticed while doing all this is that we can actually see different modes of action. So here we have, for example, 3 different strains that are exposed to the MIC, in this case of a beta-lactam, and then decreasing concentrations, increasing concentration. You can see here's the control, not exposed, and then there's a drop in cell count, and then they basically disappear. Whereas exposed to crinolones, you can see that sub-MIC, already the population extends, and we think this may be, for example, a stress response. That MIC, we do see it shift back. However, there is still a population that is higher here. So that can give us indications possibly of resistance or of heteroresistance. Whereas for aminoglycosides, essentially there is no shift in the population. All we can see is a cell count drop. So in other words, mode of action of an antibiotic affects its cell signature sub-MIC. So we're investigating further what that actually means. And the susceptibility can be defined using different metrics. So drop in cell count for aminoglycosides, whereas a shift in population may be more relevant for other antibiotics. So in conclusion, this IFAST bacterial impedance cytometry is a fast, label-free, single-cell, low-volume, low-concentration antimicrobial susceptibility test. It has very high concordance with standard methods. It works on all the bacteria we've tested so far, so it's quite fun to play with. It works for various antimicrobials, so from phage to antibiotics and so on. It also gives additional information about bacterial responses to antimicrobials, like I showed with the phage or with the aminoglycosides, and there's multiple applications. That leads me to thank everybody involved. So it's obviously a big team at UKHSA, at the university and at the hospital. And obviously without funders, nothing could ever happen. And we are currently actually advertising for a PhD, if I can just do a plug, which we will be advertising hopefully today, if not tomorrow. So I'm happy to answer any questions.
Lucy, thank you very much for that very nice presentation. I have a question, but there's a question or maybe a comment from the participant, the audience. Says preliminary findings are great, but application in low-resource countries might be difficult due to cost per test. Do you have any views on that, or?
Yes, so essentially, like I say, the idea was to have it cost the same as a current test, so the same as an agar, a disc diffusion, or a microboth dilution. So the actual, obviously, you would have to buy the machine, but even that, I mean, you'd have to talk to IFAST. I don't represent IFAST, but that was the aim of the project, and we do have a health economics study coming out. Well, it's submitted to BMJ, so waiting for the reviewers' comments that will hopefully also answer that. But that was one of the aims, so I don't think it should be in any way limiting, hopefully.
Yeah, I mean, again, I don't belong to IFAS, and I have no conflict of interest with them. But from my understanding, the technology, you just need electricity, isn't it? It's not demanding on reagents or. So it's really, if you have a solar panel, maybe in future. I mean, I have been talking to the Professor Howell Morgan in the physics division to see whether we could try it in Uganda, because it's not very demanding from the reagent point of view, and it's just electricity. And most places, or at least places where they provide healthcare, they could have generators or solar panels in those areas.
And also we work with very small volumes, which is helpful
Yeah.
also because, like I say, we're only looking at about 30 microliters in the end. So, I mean, okay, call it 200 because, you know, by the time it's flowed through, that would be the maximum, basically. So, yeah.
And one other, I know you have worked on MRSA, Neisseria gonorrhea, E. coli, Klebsiella. Is there a plan to expand to other causes of UTI, Proteus, Enterococcus, etc.?
Yes. So we currently have a student looking at both other causative organisms and also mixed cultures, because obviously UTIs especially are usually not pure, and we can actually see quite, because I was showing that different organisms will have a different signature, and very often a mixed or contaminated culture for UTI will be a gram-positive and a gram-negative, and you can, in most cases, quite nicely separate them. So you have a signature of two clouds, essentially.
So you could separate mixed
Yes.
cultures or mixed.
So I don't want to say always. We're working on it to check, you know, what the limitations are, basically. But yes.
Let me see. I think there are a couple of more. It's nice instrument, IFAST. Which method will you recommend as the best for AST?
Overall, I mean, I do think that this could change things because it is so much faster. So, I mean, one of the things is empirical prescribing of antibiotics. So, you know, if this can be done, for example, with mixed cultures and currently I said we expose for 2 hours. That's because we want to make sure that we see all the reactions for all the different organisms. But for most organism antibiotic combinations, we actually see results after 30 minutes. So we're talking the future now. But I don't see any technological reasons why this couldn't be placed, in a primary care setting to test before empirical prescription.
Yeah, I think the methodology would depend on your resources and availability of technical ability.
Yeah.
But I think it looks promising. Let's watch the space for IFAST. Lucy, thank you very much again for the presentation. Thank you. Our second talk is from Professor Jonathan Edgeworth, who is clinical microbiologist, professor of clinical microbiology at Guy's and St. Thomas's, with interest in the diagnosis and management of intensive care infections. He has a long-standing research in antimicrobial resistance, but over the last 5 years, he and his group have been increasingly focusing on evaluating the potential and the impact of nanopore sequencing in a real-life clinical setting. I think COVID played a role in this transition from AMR to sequencing and metagenomics. Prof. Edgeworth joined Oxford Nanopore Technologies on a part-time basis in October 2022. He now split his time between leading a translational research program at St. Thomas's Hospital in the UK, and he's been based also in Oxford, providing clinical insight to requirements for implementing rapid pathogen sequencing into routine lab tests. So I will hand over to Prof. Edgeworth to talk about metagenomics in clinical practice.
Thank you for that kind introduction, Kordo. So I'm going to talk about the journey from developing and evaluating a sort of sequencing capability through to a research setting and then a pilot service. Of course, that works through a lot of different organizations, your university, the hospital, your pathology provider, and the technology company itself. That's the disclosures you've mentioned already. I've stopped my clinical work, so I just do research at St. Thomas's and part-time VP Medical Affairs, Oxford Nanopore Technologies. Those are our funders and statements of the use of the technology. So, a bit like the last talk, really, and certainly for metagenomics, these are disruptive propositions for the status quo. And so I think at some point you've got to take a step back and say, well, what's the opportunity here? Metagenomics is sequencing all organisms in a sample. And so it's not just identity, it's what AMR factors, what virulence factors, what type they are. So essentially you get all the information in one go. So let's look across infectious diseases, which of course are changing, huge numbers of threats. And the sepsis doesn't need introduction. Pneumonia, which is fourth biggest cause of death, most common cause of sepsis. And there's antimicrobial resistance. We use the antibiotics to treat those conditions. Perhaps the biggest cause of death worldwide. Sorry to keep banging on about death to capture people's interest, but that is the reality, as these reports say. By 2050, if we carry on at the current rate, and then we've just had pandemics due to COVID, a respiratory infection. And also, I think there's an increasing concern about biosecurity, benchtop DNA sequencers, and AI being able to predict what the three-dimensional structure of a protein will be from a synthetic DNA sequence that could allow, bad actors, as described, to make artificial pathogens of increased virulence to provide a biosecurity threat. So that's taking things in the big sweep that can help bring stakeholders together to help us overcome the challenge of introducing disruptive technologies. And actually, when you look at that broad sweep of challenges we face, and then you look at what our laboratory networks are now, clearly, I don't think they're really well set up to meet that need. I don't know in other countries, but certainly in the UK, universities sort of detached themselves from the routine microbiology lab a long time ago. That becomes a core service. often off-site. The focus is on automation and Chiestra machines, and the separating of acute infection care decisions from public health surveillance and prevention. And there's a sort of packaging and sending of samples in a very analog way, and data sheets and typing and reference data that comes back over a long period of time. And so everything's sort of disconnected and doesn't work very well. I think COVID did drive things forward. So public health laboratories are certainly standing up pathogen genomics, so either targeted sequence of organisms, SARS-CoV-2 sequencing, flu sequencing, RSV, etc., isolate sequencing to look at AMR, and a surveillance and genomic strategy. But I'd sort of like to ask, what are our routine microlabs able to do in terms of standing up pathogen genomics to help patient care? And that's the focus of my talk today. And SARS-CoV-2 is targeted sequencing. You do an amplicon, and you say that's the only thing I want to look at in that sample. You PCR up the amplicons, get the full genome, and the man in the street, woman in the street is aware. Every time a new variant came along, that it would hustle back into lockdown and the understanding of sequence that helped us track the pandemic. But I want to talk about this metagenomics, which again is an agnostics capability where you essentially sequence everything in a sample and just talk through the lab development, the research, and then a pilot service evaluation and look to the future. And this is one side just on the technology. I think the beauty about nanopore sequencing that's attractive to micro labs that perhaps can't afford the infrastructure investment of large scale sequences is that the sequencer is only about $1,000, and there it is stuck in the side of a laptop. It's got lots of little pores, and there's proteins that grab the DNA, take them to the pore, and it goes through the pore, and there's electrical charge across there. And as the CTs, A's and G's go through, it perturbs the electrical charge that gives a signal that can then be interpreted in real time into CTAs and G's. And that's a sort of unique approach, and it's really quite amazing. You've got 400 bases a second times perhaps 1,000 pores, 4,000 bases a second all going through. So a postdoc in the lab did her PhD in the University of East Anglia and developed this 7-hour protocol where you first of all take a respiratory sample, you deplete the host DNA, you don't want to sequence that, you extract the microbial DNA, PCR amplify it, and then sequence it on these nanopore sequencers. And she joined March 2020, and while I was on the ICU, would go down and identify patients who had a secondary infection, and the clinical lab would anonymize those samples and hand them to the research lab, and they would be batch sequenced. And this has been published and managed to, in 8 hours, provide a sequence report with good sensitivity and specificity against bacterial identification. You identify beta-lactamase resistance genes, you report on those, and good concordance with Aspergillus. And then also, so you've got this respiratory sample on a sequencer and it's churning out the information. You can get a report after, 2 hours of sequencing that you can act on. But if you carry on sequencing overnight, you get enough DNA sequence to construct full genomes, and therefore you can do typing. And this was 6 or 7 respiratory samples, and most of them, this is a Klebsiella pneumoniae, most of them different by thousands of bases. They're clearly different. There's no transmission there, but there's 2 samples. There was 4 in the end that only differed by a few SNPs. And then we had 7 ICUs at the time seeing COVID patients, and all those 4 patients were on the same ICU. So you're matching the sequencing with the clinical epi. And so that's pointing to the notion of you do one sequence report, you can use it for treatment, and you can also use it for infection control. So if we jump back to winter 21-22, we were actually concerned that there might have been yet a further COVID pandemic wave and a resurgence of flu. And for those of you who are treating COVID patients on the ICU, it was hugely frustrating, very sick patients, very inflammatory, but much concern about secondary infections. But culture, of course, takes 48, 72 hours. So we fast-tracked the evaluation of real-time nanopore sequencing in an evaluation setting on the ICU, where the intensivist would identify the patients they were concerned with, give us the BAL, 8 a.m. in the morning, and then about 3 o'clock in the afternoon on the ward rounds would go down and give them a metagenomic report. And this is, just to give you some idea of what it would be. So the metagenomic sequences everything. So there's your names of all the bugs, and there's a bit of Staph aureus there and a few anaerobes. You put in a a bug at the limit of detection, and we have thresholds for reporting. So we'd report that one as Staph aureus and mixed anaerobes after 30-minute sequencing. Our 2-hour report, if you see any more 2-hour sequencing, we'll let you know. And then that's what culture said at 2 days, scanty Staph aureus, and at 5 days, no anaerobes. I mean, that's a common thing. Obviously, we know anaerobes are hard to find in the microbiology lab. So that was what was put into the notes. So we've published earlier on this year the pilot service evaluation, and this is after 2 years of service. So every winter we go into service, where a pilot service evaluation, where the intensivists choose the patients that they're most concerned about. And we do a daily metagenomic sequencing run and provide those results back within between 3 and 4 o'clock in the afternoon. And then we collect the data. We have an oversight committee that will review the results of patients every couple of weeks. And we have a mechanism reporting any adverse incidents or any safety concerns or any major errors, of which we actually haven't had any significantly, other than an unexpected Mycobacterium tuberculosis in a sample that was triggered because they were supposed to be excluded by the clinical history before anyone with a suspected Cat3 pathogen. So that's the only time we needed to communicate it. And the service has been going this winter as well. It's the last day tomorrow. We've done 107 patients over this winter. But first, a couple of hundred odd tests are reported here. That's the median turnaround time, 90% same-day reports. And the key headlines, and I think this is the link into antimicrobial resistance, is that in almost half of cases, antibiotics are changed in response to giving this information. 20% escalation, 25% de-escalation. And of course, ICU, you go down and tell them there's a bug in the chest they're not treating, they've got the pen out already and they're writing up another antibiotic. You tell them that they can de-escalate because, they're too broad or too many antibiotics, and they'll say, I'll have a think about that. They mostly do that the next day. It sounds reasonable to me. There's also benefit without antimicrobial change. Sometimes you're stopping somebody from escalating, so they're concerned, they're on antibiotics, antimicrobials. You just come back in the evening and afternoon and say, look, this is what's there, nothing unexpected, just what you've seen before, so they don't escalate. And sometimes it can help prompt immunomodulation. And sometimes you see some unusual and unexpected things, and I'll perhaps talk about that. So this is a bit of headline into the antibiotics, just to give a flavor of what those decision changes are. And actually, I suspect there's not many surprises there. When you ask to escalate, it's usually to meropenem, and that's if you get an Enterobacter or serratia in somebody who's got ontazocin like that, or linezolid because there's a VRE or a MRSA, and they're not on it. The de-escalation is pretty similar. You can stop the meropenem and de-escalate to comoxiclav or tazocin, or you can de-escalate the tazocin. And then this is the reassure group, and then in sometimes no impact because nobody made a decision, infection was at another site, etc. I want to draw out anaerobes actually and say that, as a clinical lab, we just don't detect them really, do we? So they're not on our radar. We don't think of them as cause of respiratory infection, but I think this is potentially a really helpful, important clinical benefit of metagenomics. So on the day you see the patient, you actually say, there you go, long list of anaerobes. 44-year-old woman, empyema, sore throat in the weeks leading up to it. We're concerned about strep pyogenes, high PCT, but it's not strep pyogenes, so you don't need. So they didn't need to give IVIG. The BAL didn't grow. Pleural fluid, it crawled out after prolonged culture, but we could de-escalate early on to comoxiclav with a bit of metronidazole at the start. Pleural fluids that usually do grow when you get anaerobes from a pleural fluid, they tend to grow in our experience. So empyema, reassured, confidence to continue on comoxiclav rather than escalating up to other things. And then there's a car accident here, anaerobes just weren't grown. Somebody in hospital who'd been off antibiotics for a while, anaerobes, culture didn't give us anything. So you can start comoxiclav. You don't go, more unwell, CRP's jumped, tazocin and meropenem. You go, well, look, actually it's anaerobes, and go with coamoxiclav. So I think there's lots of benefits there. This is a more targeted case where it's sort of really revealing, the breadth of opportunity you get by doing metagenomics on a patient. So this was flu, 54-year-old man, comes in very inflammatory, linezolid and meropenem as you do. BAL was taken at 10 to 2 in the morning after they'd been stabilized, and 5 o'clock that afternoon on the ward round, we could come along and tell them on a 30-minute sequence report they had PVL, MRSA, and strep pyogenes. There's the Staph aureus, there's the strep pyogenes, and there's the cutoff. You also, a lot of C, MEC-A, so that's the basis for the MRSA, LUC-FS, that's the basis for the PVL. Carry on sequencing, and the next day you can tell infection control, MRSA, MLSTs, ST152. Actually, the patients were started on IVIG in response to this. Probably would have got round to it in the end, but anyway, this prompted that. Interesting, of course, some guidelines would say you'd use clindamycin for PVL MRSA. So this allowed us to start rifampicin with some confidence. And then all the culture does at day 5 is MRSA. So this was October the 8th. We didn't really think about the strep pyogenes. And then, we had that massive increase in group A strep that year, didn't we? Around the UKHSA announced on December the 6th that there had been this massive spike in group A strep. So we said, we had 2 or 3 group A streps back in October. Now, if you then said to the routine lab, you had a group A strep in October, can you get it out and have a look at it? I think probably most labs would have chucked it away. But of course, because it's sequence data, it's all there. So we took the 24-hour sequence report and sequenced it, and it was the same variant that it was causing the infection in the children later in the year, the MM1 UK strep pyogenes with SEAJ. So that's just an illustration of the public health benefit of metagenomics. This is an ICU case, ICU acquired, 59-year-old woman who has an aortic mitral valve replacement, is extubated early, but then has secretions and high CRP, back on the ventilator, and started on Gent and Vanc because pen allergy. So I was with the intensivist that afternoon, and we said, look, let's do the metagenomics tomorrow morning. And came along afterwards and said it's Legionella pneumophila, which is really surprising. And of course, we all know about ICU-acquired Legionella, but you don't go around, most of us, I don't think, doing PCRs on every post-op patient who deteriorates. So it was urinary antigen negative, actually PCR positive. It was an ST1326, and we looked at the water taps around that part of the ICU, and bed 21's water tap and 19 there had lots of the same serotype. So serious incident declared, filters fitted, and presumably infections prevented. And then I'm just illustrating here a really key thing that's coming out from the metagenomics, because we're still trying to work out what exactly are the benefits of metagenomic service. And I think this just illustrates it in a vignette. So a 72-year-old woman with some comorbidities, bit of ear discharge on Coamox. And when the ear swab result came back around here, let's escalate to tazocin and send a sputum sample, not doing very well. When that comes back as heavy growth Pseudomonas, even though on tazocin, ciprofloxacin is added, but it's not doing very well. So we do the metagenomics and we find Pseudomonas and SARS-CoV-2, which we all know about. This was close to being in the pandemic, but had 2 negative SARS-CoV-2s. And that's the thing about metagenomics, is if you had it as testing all your samples, then every time you would look for everything. You wouldn't have to be clever. And here, many of us might have just said, it's not there, so let's not think about that. And then when we had the Mpox outbreak, cases were increasing rapidly in the UK. There wasn't a PCR test. And we have a high consequence infectious diseases unit, and we had 3 patients in at the time. We managed to get a health and safety agreement to allow us to do metagenomics on those samples. So some skin swabs were taken, and within 30 minutes of sequencing after a 7-hour run, 2 of them were Mpox and 1 of them was varicella. Very hard to tell the difference. Now, I think the PCR samples were going through that day, so it came along at the same time, but it's just illustrating the ability, of the power of metagenomics. You don't need the PCR test, it's agnostic. And we published the first full UK genomes. So I've only had 15 minutes, which is why I sort of went quite fast, and there's lots to say on all of those things. I'll just leave with where we are right now. I mean, I think we have managed to capture the imagination of NHS England and government to say, look, this should be the future. And given all of those infectious threats out there, pandemics, biosecurity, AMR, we need to expedite implementation and evaluation. Perhaps not something we can spend 15 years working its way through things slowly. So we've set up a network of UK sites that are looking to implement a similar sort of metagenomic capability as a prelude to moving into trials, improvements of the technology, and potentially commissioning. under this Network of Excellence group. And yeah, so this winter service has gone well again. I think we're seeing the same sorts of things that we did before. There's unexpected infections. The power of having an agnostic sequencing capability. It's feasible. We do 3 to 5 samples a day. It's working well, not many failures. And so I think we're encouraged that this is something that, if we keep on down this path, has a potential to translate into a routine setting. Thank you.
Jonathan, thank you for that very nice presentation. Let me see the question. I have one question. I mean, it's again related to the technology. Do you see this technology in the future to be like a black box, to be cost effectively used elsewhere, not within a highly developed country or healthcare setting? Do you think that will be possible in the future? And you're on mute. You're on.
That is the vision. That's where we need to get to. It can't just be in a few centres. It needs to be widely deployable. It is a little small device the size of a mobile phone, even some that might be fitting onto a mobile phone. The technology, the bioengineering is proceeding at an incredible pace. You know, you combine AI with microfluidics and electronics. You know, this is tractable. It's about bringing that into a box. It's about registration. It's about, you know, evaluation in the field. It's about willingness of interest from people in different settings to it. I mean, this has gone up in the International Space Station. It's been used in, you know, in the Ebola outbreaks. It's been the Zika. I mean, it's demonstrated in principle that it can work in those settings, but it's now just deploying it in healthcare settings where it literally can be sort of sample in, answer out type thing, the way the BioFire has gone. And I think that's feasible. It's just, I think, you know, you need funding, you need ambition, you need a rationale and a plan, which I think is coming together.
Yeah, thank you. One of the questions from the participants is, would lateral flow tests be quicker than genome analysis to confirm the presence of viruses?
Yeah.
I mean, I would imagine one of your COVID negative one. I know lateral flows it could be quicker, but I mean, we are always. I mean, lateral flow is good for things that we know about. I mean, the advantage of the metagenomics is we are looking at, it's like a gunshot. We're looking at unknown. We are looking for the unknown, while the lateral flow, if you know you're dealing with which virus, of course, it will be quicker and probably much cheaper. But I think there are the unknowns that we are trying to look for. And even with the lateral flows or some other PCR tests, I think you mentioned your case, which was COVID positive. Despite the PCR was negative, the metagenomics detected the SARS-CoV-2. Again, so it probably is coming down to sensitivities. And
no, I just think that they were negative at that time, but 2 or 3 days later they got it. We can't do every test every day, can we? And also, actually, infectious diseases is a detective story, isn't it? New England Journal loves having the most interesting cases, the ones where, and then finally they did a test and found out it was a weird and wonderful infection. It's the most exciting discipline in medicine, isn't it? I think we miss a lot. I think there's hidden stuff. You only know what you know. You only find the ones you find and then have the nice case report. But what about the ones you miss? We've seen enough of it now each year to know things are getting missed. You need an agnostic net. Every time you do an infection test, you've got to do the things you think of and the things you don't think of. I mean, I think lateral flows are great in emergency pathways and what have you, and out in the community and in our homes, but we need to move towards everything all the time. That's the vision anyway.
Yeah. The question says, are the results of the pathogen sequencing going to be available in the public domain along with clinical details?
Yeah. So put all of our information publicly available through that publication on those patients, anonymized, of course. Actually, that network is being set up because the data is being transferred centrally to UKHSA. We've had the first positive data flows. That will have human reads removed. It will have some metadata, and then the plan is for there's a group called MScape and UKHSA who will develop mechanisms of analysing that data and perhaps even looking at the unclassified reads to provide early warning from a public health perspective. In fact, that's the priority. So yes, so that's the intention, that once you've got the data, it can be immediately sent anywhere it needs to be sent. So, if the EU wants, the antimicrobial resistance survey, rather than asking a laboratory to just say how many invasive isolates have you had that you've reported in your lab as resistant, you can now send immediately, rather than it being done every year and it being static. All data could be shared, whichever organization you get the governance approvals for whatever it wants to be able to track in real time. And of course, it's not just carbapenem resistance. It's, well, what about if it's a certain mobile element, a certain new CTXM whatever, or a CRE whatever? You get all that richness as well, and you get it immediately. That's the thing. So you bring your infection control team and your antimicrobial stewardship team and your people treating patients all to be thinking about the same problem at the same time, rather than one of them thinking at one and then a week later you have another piece of information in a different room. It's really bringing us all together.
Thank you. There are 4 more questions, at least, on the chat, but 3 of them are related. And they are about, is it discriminatory for spurious result based on temporality of infection? Example, COVID, I would assume they say COVID RNA 10 in up weeks post-infection. And how would whole genome sequencing is able to differentiate contaminants from clinical pathogens? And the last question related to this topic says, excellent talk, the presence of few pathogens in the metagenomic data. Am I correct to say that it still needs some clinical interpretation to decide antibiotic escalation, de-escalation?
Totally needs clinical interpretation. It is ultimately still telling you there's a bug there, but we then need to see the patient. So totally still needs clinical interpretation. And the reports need more work. And the bioinformatic tools need improving. There's improvement all the way down the line. First question, really pleased to say that the method that is employed here removes at a critical point. DNAs will digest all the human DNA, and it digests all the free microbial.
Oh, I think we lost Jonathan. I think Jonathan was trying to say to deplete human DNA, and then once you have detection of pathogens from the metagenomics, I think like any other results, you have to interpret it within the clinical context of this patient. I hope we will get Jonathan back to answer. There's one last question on the chat. Okay, I think we've come back to Jonathan. Hopefully we'd be able to come back. But I think most of his question is answered. Is something related to cost effectiveness in low and middle income countries. And at the moment, we know some of this technology can be costly and can be demanding for expertise. But I will let him to feedback if he was able to join us again. And there's one comment or question about the clinical trial was recently expanded to more NHS hospitals. Have these been installed yet? We are currently in Southampton. We are one of those sites, and we are hoping to start the metagenomic sequencing we have done our extraction. We haven't done the sequencing yet. So hopefully it's on the way for the next winter in the UK at least. It looks like Jonathan might be having trouble with his connections and IT, but we have our last speaker who's patiently waiting, Dr. Saranga Sumathipala. He is a clinical virologist and head of Department of Clinical Virology and co-facility head, Next Generation Sequencing Lab at the National Cancer Institute at Kisha Hospital in Sri Lanka. I'm delighted Dr. Saranga is with us today. He studied medicine at the University of Peradeniya and received his postgraduate diploma in medical microbiology from the University of Colombo. He's a board certified as a specialist in medical virology in Sri Lanka. His areas of expertise are lab diagnostics and medical microbiology, virology, molecular diagnostics. And he's here today to talk about a rapid diagnosis of hepatitis B in the low and middle income countries and impact on preventive measures. So thank you, Dr. Saranga, for joining us. The floor is yours.
Thank you, Kordo, for the kind introduction. The disclaimer, I'm an employee of Ministry of Health Sri Lanka, and I do not have any external affiliations, nor funded externally, and my presentation is based on experience in Sri Lanka. So what about hepatitis B, the virus, the global burden? According to WHO, it's a hepatotropic virus which may lead to chronic infection with sequelae of cirrhosis and hepatocellular carcinoma. According to WHO, there is an estimated 254 million people who live with Hep B virus infection, and 6,000 people are newly infected with viral hepatitis each day. And the bulk of infection burden is been seen in the low and middle income countries. On average, 3,500 people die every day from hepatitis B and Hep C combined. And the sad story is only 30% of people living with chronic hepatitis B infection had been diagnosed, and close to 3% had received antiviral therapy at the end of 2020. So majority is undiagnosed. Just a little bit about Sri Lanka, where I come from. Sri Lanka is an island nation in the Indian Ocean. It's very close to the equator and has a population of 22 million, and is a lower middle income country. And it's a small island, total area of 65,000 square kilometers, and anyone can travel from one place to the other within a day, probably within half a day. So it's important to say, because we do transport specimens from one side to the other side. What about the hepatitis B, the Sri Lankan status? So just a bit about our healthcare system. It's a universal healthcare system managed by Ministry of Health of Sri Lanka, and there's a smaller, thriving private health sector. Regarding hepatitis B, the prevalence in Sri Lanka is less. It's a low prevalence country. Fortunately for us, it's been supported by the numbers are supported by serosurveillance data and data from the National Blood Transfusion Service from the donor surveillance data. And what about the diagnostic facilities of Hep B? Just to show around. So we have limited virological diagnostic facilities in the country. We have comprehensive virology services by regional reference laboratories. So one at Anuradhapura, Kandy, Maharagama, and Karapitiya. And there's a central, the reference lab, the Medical Research Institute. So these are the 5 virology diagnostic facilities in Sri Lanka, which offers exclusive total screening and confirmatory and follow-up facilities, including serology and PCR. And there is limited hepatitis B screen capacity in district clinical microbiology labs, so which does conventional serology, and some of them can do surface antigen and hepatitis B surface antibody levels. And of course, we do have a ongoing routine screening of hepatitis B surface antigen in blood donors by the National Blood Transfusion Service, which is in decentralized places in each hospital where blood transfusion services are centered. And of course, there are private sector laboratories, which is limited and localized to a few major cities, but they have very good logistic service from collection centers all around the country. And a few does comprehensive hepatitis B virus serology plus PCR. And we do have therapeutics and vaccines available centrally in major hospitals, but can be arranged for any patient on demand. And of course, we do have a well-functioning and successful hepatitis B virus elimination program targeted through National Childhood Immunization Schedule, the EPI program, where we vaccinate children universally at the ages of 2, 4, and 6 months. But we do have limitations and challenges in diagnostics of Hep B. The major 2 challenges are the limitations of laboratory infrastructure for biological diagnostics, and human resources, because our numbers of expertise, especially the laboratory scientists and the laboratory consultants, are very limited. And of course, there are issues with test and reagent availability, because I mean, as you have known, we faced economical meltdown a couple of years back. We had gradual recovery, but despite that, we were able to maintain fairly stable level of reagents for our diagnostics. And of course, there are delays in specimen transport to labs, and there are logistic issues. And there's always a necessity for an actionable diagnosis, especially for screening of hepatitis B in the Sri Lankan population. So just to show you, the turnaround time for Hep B diagnostics, which is from sample to result in our country, rapid test, whenever there are, it takes minutes to hours. And we do have serology, comprehensive serology, CLIS and ELISAs. It may take hours to days, depending on delays in sample transport. And for example, especially for ELISA, we do batch testing. So sometimes the TAT is counted in days. And we do have molecular and standard qPCR assays, which will range from the TAT will range from hours to days. And regarding confirmation, we always follow up with the confirmation of hepatitis B when there is an initial screening positivity detected. For that, again, depending on the delays in logistics, the TAT would be from hours to days. And sometimes it may take even weeks in certain situations. For example, in the National Blood Transfusion Service, because samples were collected and sent in batches to the reference virology labs. So just to show you this is like any other testing algorithm. This is a general testing algorithm, the workflow for hepatitis B screening. We basically rely on hepatitis B surface antigen plus or minus co-antibodies depending on the clinical scenario. And if it is reactive, then we follow up with a second sample. We send a sample to a virology lab for confirmatory testing. And of course, we do follow up testing. And when do we do hepatitis B virus testing in Sri Lanka? Basically, as I mentioned, for bloodborne virus screening, we do 5 pathogens. One is Hep B. Antenatal screening is not universally done, but especially in the private sector for EMTCT elimination of mother to child transmission of Hep B, there's a concern, and they do Hep B screening. And of course, we screen high risk and high prevalence groups. For example, people with STIs, high risk behaviors for Hep B, such as injecting drug users, and also patients being hospitalized for oncology. For example, I work at the National Oncology Cancer Institute. Our institutions are basically crowded, and when the patients they are given intravenous, there's a lot of intravenous access going on, and so like many years back, probably a decade back, for example, we used to have a lot of Hep B, and we now have established a screening program for all the oncology patients, and then we follow up with vaccination. And of course, contact screening of confirmed and non-Hep B patient is also being done. And in the clinical setup, patients with clinical hepatitis, whenever there is a needle stick exposure, needle stick injury, or a BP bloodborne virus exposure, we do screening. And of course, we have confirmation and post-emergence antibody testing, which is again mostly confined to the virology labs, which I mentioned earlier. But the role of hepatitis B rapid diagnostics, or RDTs, so it basically broadens the access for testing and facilitates testing for screening with the shortest TAT for decision making, and it provides a decentralized access to testing at point of care. And only as we are aware, there are only 2 targets being offered for commercial assays, nominally B surface antigen, and there are point of care tests which offers hepatitis B quantitative viral load testing. What about the rapid diagnostic test for hepatitis B surface antigen? Basically, they are, majority of them, almost all of them, are lateral flow assays, which are designed for portability and point of care testing. The advantages, it can broaden the access to diagnostics, and technical less demanding, and it's very helpful when there is underdeveloped infrastructure. Plus, you don't need a laboratory scientist to perform the test. And the turnaround time is less for practical decision making. What are the disadvantages? It's a major drawback in the RDT, the lateral flow test for surface antigen testing, is the sensitivity, which we are again concerned, but we basically use them. So sensitivity is, according to one study done in our country, is like 60 to 80% when compared with the standard serological testing. And also, when you get a marginal result, the interpretation would be subjective. For example, faint band could be interpreted as a negative by one person and a positive by the other person. And of course, there are QC issues. You cannot standardize quality control because it's subjective assessment. What about selection of a lateral flow test? So, as I looked up, there are several lateral flow assays being recommended by WHO, such as the Determine and the Bioline. And the situation in Sri Lanka, so we do get testing available in Sri Lanka from time to time. So the products are like the brands are variable, and we always do verification of a new test kit locally before giving the green light for procurement for the national level. And the latest I could find is this brand called On-Site Hepatitis B surface antigen combo rapid test by CTK Biotech. The rapid test for surface antigen, when and where do we use it? So the preferred sample is peripheral blood or serum, and we have it in some of the district laboratories. Just want to mention it's not available universally in all the microbiology laboratories, and we try to use it where there is some sort of laboratory expertise. That's why at least I confine it to the microbiology labs. And the other advantage, we are less concerned because our prevalence is also less, so we don't use it throughout everywhere. Basically, for needle stick injuries and bloodborne virus exposures, we use it. And of course, again, even in the regional reference virology labs, again for the same purpose, we use the rapid test in parallel with conventional serology. For example, when a particular reagent is not available in the lab, we rely on the other. So sometimes even we may have to resort to rapid tests, but we use them concurrently. And there's a discussion being carried on at the moment about triple testing for HIV, VDR, and Hep B as a national program for elimination of mother-to-child transmission. So at some point we may introduce it, the rapid test, to be used for maternal screening as well. So when do we not use it? The rapid test for Hep B surface antigen, inconclusive results, faith bands. When we receive one, we would not use it. We would send a sample straight away for conventional virology testing. And if a patient is having incompatible result with the clinical history, for example, if a patient is highly suspicious of having hepatitis B virus or some sort of a chronic hepatitis, yet the result is negative, we will definitely send the sample to a virology lab. And patients with clinical hepatitis, again, even for full-blown hepatitis, we would prefer to use the conventional testing pathway, because another reason is because we want to test them comprehensively for other hepatitis viruses as well. And of course, immunocompromised patients, we do not want to use surface antigen RDTs because of sensitivity issues. And again, context of non-Hep B patients also, we would like to test them comprehensively with other targets. Therefore, we prefer the conventional testing pathway with conventional serology over rapid test in these patients. And what about molecular testing? We do have molecular assays, the conventional PCR, but there are point-of-care PCR platforms available, which are random access for sample testing in self-contained modular automated nucleic acid with automated nucleic acid extraction, PCR, and reporting platforms. Everything is automated, like more or less. The good examples are the Cepheid Gene Expert platform and the Mobile Diagnostics platform. It's named the TrueLab platform. So, what about the Gene Expert platform? Preferably, it's a good platform, but it's expensive in countries like us because Gene Expert basically we use it for tuberculosis and HIV diagnostics because it's given at the cartridges, the reagents are given at a concessionary price uh, from the manufacturer through various funding sources. But it's hepatitis B is not covered by this HBDC access program. So it's relatively very expensive when you compare with the conventional, the routine PCR testing. The other platform, Mobile Diagnostics, is a platform in which I'm also using in parallel to my standard qPCR test. It's a bit expensive than the standard qPCR test, but since the prevalence is less, we do like having reagents exclusive for qPCR. I mean, sometimes the reagents tend to stay there and expire in the lab and tend to throw it out. But if we have a lesser number of modular testing, so it's more practical and cost effective. So we do have this mobile platform in parallel with the other assays. So conclusively, impact of rapid diagnosis test on preventive measures. So just to say, the most useful RDTs are the lateral assays for hepatitis B surface antigen. And the biggest advantage is broadening the access to testing in a resource limited setting. It's very useful in some in same day diagnosis when the patient is in a clinical setting then and there, especially for outpatients, because sometimes a patient comes and the patient is lost to follow up. So it's really helpful if we can test the patient then and there, and so we can give a verdict and we can ask the patient to come for the follow up. And invariably, Hep B surface antigen reactivity is escalated for confirmatory testing with the second sample and sent away to a virology lab. So we don't stop there. We always send the second sample for identity confirmation and confirmation of the diagnosis. And confirmed positive results are referred to a patient management pathway, either locally at the same hospital if the clinical expert is there, or at a tertiary care hospital nearby. And it was broad access to diagnostics. And it helps in early intervention for prevention of progression of disease, early intervention, prevention of transmission, and early involvement in contact tracing and screening. So basically, that's I wanted to present. I would be very happy to take any questions there are.
Thank you, Dr. Saranga, for this nice presentation on rapid diagnosis of hepatitis B and impact on prevention measures. You mentioned lateral flow tests, and you mentioned Sri Lanka is a low prevalence country.
Yes.
Do you have a service that for remote areas of Sri Lanka? Do you have a service of mass vaccinations in remote areas? And how do you do it? Do you just go because you are low prevalence? Do you just go vaccinate?
Yes. So basically, we vaccinate the high risk groups. So, for example, if there is someone with the cancer coming to us, so I hope you are not disturbed by this thunderstorm outside.
Oh gosh, yeah. I wondered what that noise was.
Yeah.
Hope you're okay.
Yeah. So it's normally the tropics in these days. So just to continue with the discussion, so we do have vaccination available. Basically, vaccination is mainly covered in the national program of child immunization. and we do have adult vaccination available. We do vaccinate healthcare workers. So that's number one. And people who are at high risk. So we have a kind of a very comprehensive network of hospitals and health institutions, various levels, starting from the primary to the tertiary level, and it covers the entire country. So if there's a requirement, basically we send the patient to a place where the vaccine is, basically somewhere at a secondary or tertiary level. So it's there, and if we don't have the vaccine, then there are plans for procurement, and so again the logistics are maintained centrally. So we can send from one place where the vaccine is to the place where the patient is.
And I've been in areas in high prevalence. I mean, I call it high prevalence. 8 to 10% of people populations being infected with hepatitis B. But there's a mass vaccination going on, but no testing. What's your view of that? Because there's no lateral flow testing, just mass vaccination. What's your view?
Mass vaccination could help to a certain extent, but if the risk is high, because in my experience, we always, if the patient is at high risk, we always look for the immune response to see whether the patient has developed adequate antibody levels. And if the prevalence is high, other thing, and the patient is infected, again, the vaccines won't do much help. So I think screening is very important. So it should go side by side with the vaccination.
Thank you. I think there's a question from a colleague here. He says, do you have a link for distributing companies? I would assume they mean companies for lateral flow tests.
So basically how this works is the companies, they have to get registered with the National Regulatory Authority. The NMR is the analog of MHR in UK. And then we do have these, we have these procurement tenders where the government, the Minister of Health, they go for large-scale tenders and they do the bidding. So everything comes through a local agent in the country. So that's how it works.
Yeah, and is the WHO website, do they recommend any, or do they have provided any details about lateral flow and rapid testing?
Yes, there are like a couple available mentioned, and yes.
So someone could go to the WHO website and try to find out which companies they are.
Yes.
See if there are other questions. One more question. Do you think we will eliminate hepatitis B at some point in the near future?
If you can achieve mass vaccination, I think the vaccination is the golden opportunity. Of course, you have to do testing. So it depends on the background. In a country like Sri Lanka, yes, most probably because it's easy to achieve that.
That's great, thank you. I don't think there are other questions. Thank you very much, Dr. Saranga.
Thank you.
Professor Edgeworth, Jonathan Edgeworth is back with us. Thank you very much for coming back. They were trying to talk about depletion of human DNA from the metagenomics, and also if you deplete non-viable organism. Did you want to continue what you were trying to say, Jonathan, or I think that's what you were trying to say. There's because the question was, how do you differentiate non-pathogen nucleic acid from pathogen nucleic acid? And I think you were trying to say that there will be depletion of human DNA and also non-viable organisms.
Yes.
Yeah.
Yes. You have to have a method to deplete human DNA. Also, you just be sequencing Homo sapiens, vast excess. So, of course, in any of these depletion approaches, the concern is that you remove some of the intact pathogens, and that's why we're trying lots of different methods to selectively deplete the human or to concentrate the pathogen, and also free pathogen DNA and RNA knocking about from dead bugs. That's all goes in the system. That's not recorded. And then, of course, distinguishing between commensals and the pathogen, that's a clinical decision, and that might be helped by having an immune marker alongside that, particularly if the differentiation of immune signaling and transcriptomics becomes to some extent pathogen specific, i.e. gram positive, gram negative, and what have you at a high level. So there's lots to do in the future. This is not a complete ready to go technology, but the progress is fast and encouraging. Thank you. Sorry to have dapped out. That was my Wi-Fi.
No, thanks for coming back to be with us. And there was one question, I think it disappeared from, you might have answered it. It was about cost effectiveness or cost of metagenomic sequencing per test. You probably have answered it, yeah.
Well, it's just to say, health economic studies need to be done. Consumables are running around in the UK £100 per sample, but it differs when you multiplex 8 on a flow cell, but it differs in different countries. And again, that's something that just needs to be optimized, doesn't it? So it's a journey on bringing costs down.
Thank you very much. Let me just check. There are no more comments or questions in the Q&A. So, and we are at 2:22. So it just gives me a pleasure again to thank our eminent speakers, Dr. Lucy Bock, who talked about rapid diagnostics in UTI, especially around AMS, rapid antimicrobial sensitivity for Klebsiella and E. coli. And Lucy mentioned it has a huge potential in the future, especially if we expand the repertoire of the organism. Prof. Jonathan Edgeworth talked about metagenomic sequencing in real-life clinical settings, especially in critical care, and impact on patients' management, specifically around AMR, early diagnosis of infection, but also infection prevention, and also about the importance of public health. And then Dr. Saranga Sumathipala from Sri Lanka talked about how important rapid diagnostic is and for prevention of hepatitis B. And he's very optimistic that we would hopefully one day eradicate, at least in Sri Lanka, eradicate hepatitis B infection. Thank you very much to you all speakers. And I would like also to thank ISAC and Fee Johnston for her support in organizing the meeting. Please don't forget to look at the ICC 2024 in Istanbul in November. It's really an exciting meeting and very good for presenting new technologies. And Istanbul is beautiful. If you haven't been there, try to come to Istanbul and join ICC meeting. So thank you, Fee, for all your support. And thanks to all participants for all their questions and comments and participations. They have been very active, actually. It was really, really a pleasure to see those questions coming through. So thank you very much all. Take care of yourselves and see you next time. Take care.